In a monastery garden in the 1850s, a monk started counting peas. Wrinkled versus smooth, green versus yellow, thousands of them, over 8 years. Nobody around him understood why. He'd just discovered the rules that govern heredity in every living thing, a century before anyone knew what a gene physically was. That monk was Gregor Mendel, and his monastery in what's now the Czech Republic gave him something rare for the 1850s. A quiet garden, total intellectual freedom, and absolutely no idea he was about to found an entire science. This is Mendel himself, the Augustinian friar whose day job had nothing to do with genetics at all. He simply had a fascination with plant breeding and the patience to track thousands of individual pea plants across generations. Think of what he was doing like this: crossing a smooth pea plant with a wrinkled one, then carefully counting exactly how many of the resulting offspring came out each way, generation after generation, until a pattern emerged that no one had ever described in numbers before. The pattern he found was strikingly consistent. Cross 2 hybrid plants, and roughly 3 out of every 4 offspring showed the dominant trait, while 1 in 4 showed the recessive one. A clean 3-to-1 ratio, appearing again and again, no matter which trait he tracked. Boil his life's work down, and Mendel actually established 3 separate principles at once. That traits come in dominant and recessive forms, that each parent contributes 1 copy that segregates independently, and that different traits are inherited independently of one another. Here's the part that should genuinely surprise you. Mendel published this work in 1866 in a small regional journal, and it was essentially ignored for 35 years. The man who discovered the rules of heredity died in 1884 without ever knowing he'd be called the father of genetics. It took until 1900, 16 years after his death, for 3 separate botanists to rediscover his work independently and realize what they were looking at. Sometimes the biggest discoveries in science have to wait for the rest of the world to catch up. While Mendel counted peas, a Swiss physician working in an entirely different city was about to isolate the actual molecule Mendel's rules were describing, without having any idea that's what he'd found. This is Miescher, and his source material was genuinely unglamorous. He was studying white blood cells, and the cheapest, most abundant source he could find was pus scraped from used surgical bandages collected at a local hospital. In 1869, he chemically separated something strange from those cell nuclei: acidic, rich in phosphorus, and unlike any protein or fat he'd ever characterized. He named it nuclein. We call it DNA. Miescher had no way of knowing he was holding the molecule of heredity itself. That's the strange shape of this story. The physical molecule was isolated in 1869. Its actual role in heredity wasn't confirmed until 1944. For 75 years, scientists studied DNA's chemistry without knowing it was the very thing Mendel's rules were describing. It's a pattern worth noticing early in this story, because it repeats. Mendel's rules sat unread for 35 years. Miescher's molecule sat misunderstood for 75. Scientific insight doesn't always arrive with its significance attached. 2 men, 2 discoveries, both essentially invisible in their own lifetimes. It would take a new generation, armed with microscopes and chromosomes, to start connecting the dots. By the early 1900s, biologists had spotted thread-like structures inside dividing cells called chromosomes, but nobody had proven they actually carried Mendel's mysterious hereditary factors. That proof came from one scientist and an army of fruit flies. This is Thomas Hunt Morgan, working out of a cramped Columbia University lab nicknamed the Fly Room. Milk bottles stacked with breeding fruit flies covering every surface, and Morgan himself initially skeptical that chromosomes had anything to do with heredity at all. Fruit flies turned out to be the perfect tool. They breed in about 10 days, cost almost nothing to keep, and one day, Morgan noticed a single male fly with white eyes instead of the usual red. A visible mutation he could actually track across generations. Tracking that white-eyed mutation across thousands of offspring, Morgan found it was inherited alongside sex, passed differently to male and female offspring in a pattern that could only be explained if the trait lived on a specific chromosome. His own skepticism had just been disproven by his own data. This was the missing link the whole field needed. Mendel's abstract mathematical factors were now physical, visible, located on actual chromosomes you could see under a microscope. Genetics stopped being pure statistics and became biology you could point to. Morgan received the Nobel Prize in Physiology or Medicine in 1933 for this work, the first Nobel ever awarded for research on the fruit fly, an animal that's since won that distinction, indirectly, several more times over. For context on Morgan's tiny research subject, a fruit fly's entire genome is a fraction of the size of a human's, small enough to work with in the 1910s, yet complex enough to reveal a real principle of heredity. Genes now had an address. The next question, the one that would take another 3 decades to answer, was what genes were actually made of. By the 1940s, most scientists assumed protein must be the material of heredity. It seemed complex enough to carry the instructions for life. DNA, by comparison, looked too simple to matter. One quiet experiment was about to prove almost everyone wrong. This is Oswald Avery, working at the Rockefeller Institute in New York with colleagues Colin MacLeod and McClintock, a careful, cautious researcher who, by his own account, almost didn't believe his own results at first. The experiment used pneumonia-causing bacteria that came in 2 forms, a harmless rough strain and a deadly smooth strain. Something in dead smooth bacteria could permanently transform the harmless strain into the deadly one. Avery's team set out to find exactly what that something was. The result pointed straight at DNA, but the scientific community's assumption ran so deep that many colleagues simply refused to believe it. Surely, they reasoned, something must be contaminating the sample. Surely it couldn't really be DNA. Avery himself was cautious in a letter to his brother, writing that if he were right, it meant nucleic acids weren't just structurally important, but functionally active substances determining the biochemical traits of cells. He undersold, in a private letter, what may be the most important sentence in 20th century biology. Confirmation of DNA's role came alongside a second crucial clue from biochemist Erwin Chargaff. Measuring DNA from many species, he found the amount of adenine always equaled thymine, and guanine always equaled cytosine. A pairing rule nobody could yet explain. Worked as a real ratio, DNA from an organism might show 30% adenine and 30% thymine, 20% guanine and 20% cytosine, always paired, always equal, in species after species, like a hidden mathematical rule waiting for a structural explanation. Zoom out across this whole story and a pattern jumps out. Nearly every major discovery so far waited years, sometimes decades, before it was fully recognized. Mendel waited 35 years, and as we're about to see, one later scientist would wait even longer. DNA was now confirmed as the genetic material, and a strange pairing rule sat waiting to be explained. 2 puzzle pieces, both sitting on the table by 1950, and about to be assembled by 2 young scientists in Cambridge. Solving DNA's actual 3-dimensional shape became one of the great races in the history of science. And the person who captured the single most important piece of evidence has, for decades, been the least credited. This is Rosalind Franklin, a rigorously trained X-ray crystallographer working at King's College London, whose meticulous technique produced the sharpest DNA diffraction images anyone had ever captured. In May 1952, after exposing a DNA fiber to X-rays for over 60 hours, Franklin's student Raymond Gosling captured an image that would become one of the most famous photographs in the history of science. This is photo 51 itself, the actual X-ray diffraction pattern. The distinctive X shape was immediately recognizable to a trained crystallographer's eye as the unmistakable signature of a helical structure. Quick pause. If this breakdown is helping you, take a second to hit like and subscribe. It genuinely takes a lot of effort to make videos like this, and your support keeps them coming. Without Franklin's knowledge or permission, that image and her related data were shown to James Watson by her colleague Maurice Wilkins. Watson recognized the pattern instantly, and it became the missing piece he and Francis Crick needed. Within weeks, Watson and Crick built this, a double helix, 2 strands winding around each other, held together by base pairs following exactly the rule Chargaff had found. A always pairing with T, G always pairing with C. Watson, Crick, and Wilkins shared the 1962 Nobel Prize for this discovery. Franklin had died of ovarian cancer in 1958 at just 37, and Nobel Prizes are never awarded posthumously, so she was never eligible to share in the recognition. Historians today generally agree the discovery genuinely required all 4 contributions: Franklin's rigorous X-ray data, Wilkins's collaborative access to it, and Watson and Crick's model building insight. A collaborative achievement. However unevenly, it was credited at the time. This isn't the only moment of contested credit in this story. Keep an eye out, because it happens more than once across this history, in different forms. Grading this honestly, as historians of science now do, Franklin's data was demonstrably essential to the discovery, used without her consent, and for decades she was written out of the popular telling of the story entirely. A wrong that's been actively corrected only in recent decades. The shape of life turned out to be a double helix, and the story of how we found it is as much about who got remembered as it is about the molecule itself. While the double helix made headlines, one scientist working alone with corn plants had already discovered something so far ahead of its time that almost nobody in her field believed her for nearly 40 years. This is Barbara McClintock, photographed in her laboratory at Cold Spring Harbor in 1947. Studying the colorful, unpredictable patterns of pigment in Indian corn kernels, patterns that didn't follow Mendel's tidy rules at all. Her explanation was radical. Certain genetic elements could physically jump from one location on a chromosome to another, switching nearby genes on or off as they moved. She called them controlling elements. Today we call them transposons. The genetics establishment in 1950 simply couldn't accept it. Genes were supposed to sit in fixed locations. McClintock's papers were largely dismissed or ignored. She responded by, essentially, stepping back from publishing for years, continuing the work regardless. It took molecular biology catching up in the 1970s and 80s, when moving genetic elements were confirmed directly in bacteria and other organisms, for the field to finally recognize what McClintock had shown decades earlier in corn. In 1983, Barbara McClintock received the Nobel Prize in Physiology or Medicine, unshared, one of very few scientists in history to win a Nobel Prize entirely alone, for a discovery the field had spent decades refusing to believe. Her legacy turned out to be far larger than corn. Transposons, the jumping genes she first described, now make up roughly 45 to 50% of the entire human genome. Being right decades before anyone believes you is one of the loneliest positions in science, and one of the most quietly heroic. Knowing DNA's shape was only step one. The next generation of scientists had to figure out how to actually read the code written inside it, and eventually, how to copy and even rewrite it. Through the 1960s, researchers including Gobind Khorana and Marshall Nierenberg painstakingly worked out the genetic code itself, which three-letter sequences of DNA, called codons, correspond to which amino acid building blocks of proteins. The full code, once decoded, turned out to be beautifully simple. Just 64 possible three-letter codons, translating down to 20 amino acids, the entire biological vocabulary every living organism on Earth shares. All of this, the code, the sequencing, the copying, sits on top of one organizing principle molecular biologists call the central dogma. Information flows from DNA to RNA to protein. Once the code could be read in principle, someone needed to actually sequence real DNA, letter by letter. This is Frederick Sanger, whose method for doing exactly that, developed in 1977, would define genetics for the next 3 decades. Sanger achieved something only 4 other people in history have managed, 2 separate Nobel Prizes. The first in 1958 for sequencing the amino acids of insulin, the second in 1980 for DNA sequencing itself. The final piece of this chapter's story belongs to Kary Mullis, who in 1983 invented PCR, a technique to copy a tiny piece of DNA into billions of identical copies, by cycling it through heating and cooling in the presence of the right enzyme. Mullis famously said the idea came to him while driving along a California highway one night in 1983, a technique so simple in concept and so powerful in practice that it now underlies everything from COVID testing to forensic crime labs to ancient DNA research. By the 1980s, humanity could read the genetic code, sequence real DNA letter by letter, and copy any piece of it at will. The stage was set for the biggest project biology had ever attempted. In 1990, an international team set out to do something almost unimaginable: read every single letter of the entire human genetic code, all 3 billion of them, from start to finish. The Human Genome Project, led by Francis Collins with a competing private effort led by Craig Venter, finished a working draft in 2003. 13 years of work and roughly $3 billion to read the complete instruction manual for a human being. The cost curve since then is almost unbelievable. That first genome cost roughly $3 billion. Today, sequencing an entire human genome costs a few $100s and takes about a day, a million-fold drop in barely 2 decades. The most recent chapter belongs to Jennifer Doudna, shown here, and her collaborator Emmanuelle Charpentier, who in 2012 turned a bacterial immune system called CRISPR into a precise, programmable tool for editing DNA in any living organism, including humans. CRISPR-Cas9 works almost like a word processor's find and replace function for DNA. A guide molecule locates an exact genetic sequence, and an enzyme cuts it precisely at that spot, allowing scientists to disable, correct, or replace it. Doudna and Charpentier received the 2020 Nobel Prize in Chemistry for this work, and notably, it was the first Nobel Science Prize ever awarded to a team of 2 women only, without a single male co-recipient. Looking back across every story in this video, the same 3 things eventually brought recognition: independent confirmation by other scientists, new technology that could verify the original claim, and simply enough time for the field to catch up. Laid end to end, the whole arc runs remarkably fast once it gets going. Mendel in the 1860s, the double helix in 1953, the full human genome in 2003, and programmable gene editing by 2012. The pace of discovery kept accelerating with every decade. From a monk quietly counting peas in a garden to a tool that can rewrite the code of life itself. This entire story took less than 2 centuries, and the people who built it were, almost without exception, dismissed, delayed, or undercredited before the world finally caught up. Here are the key references and sources for this video. Thanks so much for watching. If this helped, like the video and subscribe for more clear, simple explainers, and I'll see you in the next one.